Hello, and welcome to part 7 of Innate Immunology. This is phagocytosis. What's going on there? Hello, and welcome to our lecture part 7 on innate immunology. Here we will be talking about phagocytosis. So phagocytosis is a process in which bacteria or cell debris cleanup is going to occur around the cells, and phagocytic cells are responsible for that cleanup process. Phagocytic cells include neutrophils, B cells, dendritic cells, macrophages, monocytes. These are all phagocytic cells. They take in cellular debris or microorganisms, viruses, pathogens, old proteins, anything that needs to be cleaned up and kind of recycled. And it occurs in 6 basic steps. The first is chemotaxis. Chemotaxis is a response by these phagocytic cells to a cytokine. In this case, we're looking at C5a. We just got done in the last unit talking about complement, and C5a is a very strong chemokine or cytokine. And in this case, it's called a chemoattractant or chemokine. And this makes it a very strong anaphylatoxin because it induces inflammation through that attraction of phagocytic cells. So C5a in the bloodstream is going to draw phagocytic cells towards it. And on the surface of the microorganisms, because of the complement cascade that we just studied, the microorganisms are covered in C3b. So this is a close-up right here in step 2, recognition and attachment. You can see here the same picture that we saw when we were looking at the complement effector function of opsonization. And in opsonization, C3b is attached to the surface of this microorganism here. And so it's coated in all of this C3b, and the phagocytic cells have receptors for that C3b. When they bind to those receptors, this is known as receptor-mediated endocytosis. You probably learned about that in anatomy. And that will now carry out when they bind enough receptors and begin building this endosome. This begins step 3, or engulfment, when the phagocytic cell is actually taking in and forming a vesicle around that microorganism. In this case, in our example, a bacteria. Once inside of the what we call the phagosome, a phagocytic cell that creates a vesicle surrounding this bacteria, that's referred to as a phagosome. It's the same thing as an endosome, just occurring in a phagocytic cell. This phagosome will begin merging with lysosomes. The cell has lots and lots of lysosomes, and the phagosome matures, fully forms, and full maturity is when we see here, right? We have these lysosomes that are releasing their hydrolytic enzymes into the phagolysosome. These enzymes are activated. They're called acid hydrolases. They are activated in an acidic environment. So when these lysosomes release their enzymes into this phagosome has become fairly acidic. And these enzymes get activated, and they're responsible for breaking down this bacteria. So they start breaking down the peptidoglycan layer and any lipids, genetic material, anything that gets released by this cell gets broken down into a bunch of pieces. So that's destruction and digestion. Ultimately, we end up with all of these little pieces here. And for most phagocytic cells, this is all waste product. So at the end, we have exocytosis, where the cell gets rid of any debris that it could not break down. Now, this process in phagocytosis can take a detour. Here in the destruction and digestion stage, instead of the entire microorganism being broken down and released during exocytosis, pieces, some of these pieces may be retained by the cell and hook up with something called an MHC. And together, those are going to go out onto the surface of the cell, and the MHC is going to present. And this process here is where antigen presentation would come into play. So you do need to know the 6 stages of phagocytosis and what's occurring in each. Now, macrophages and neutrophils are the two dendritic cells as well, are two very, very highly phagocytic cells. Neutrophils are considered the most phagocytic cells in the body, or in our complex of blood cells. The macrophage is a routine phagocytic cell. This guy roams around in tissue. Remember, macrophages are mature forms of monocytes. And monocytes that move into tissue become macrophages, and they will roam around in tissue and they phagocytize everything. They are not selective. Dendritic cells tend to be a little more selective to something foreign, whereas macrophages are cleaning everything up and they are running around sampling. They also, remember, macrophages also have TLRs, RLRs, and NLRs as well. So they routinely phagocytize not just invaders, but actual dead host cells and cell debris. They are antigen-presenting cells, so remember, they have those receptors like TLRs. They can be activated to become more prolific in activity. So macrophages are antigen-presenting cells, but after antigen presentation, they will receive a signal from a T cell, or even prior to without antigen presentation. T cells will sometimes, in adaptive immune responses, find a macrophage and activate it. And depending on what chemical signal the macrophage gets, it can become either an M1 or an M2. And in M1 is kind of like the cell goes from macrophage to super macrophage and becomes bigger. Its membrane begins to ruffle. So in order to increase its surface area, with an increase in surface area, it can increase how much it can phagocytize. It has much greater killing power, has greater increased phagocytic activity, and it also releases cytokines in the inflammatory response. If it becomes an M2, a macrophage 2, it actually will regulate or downregulate inflammation, as well as release things like colony stimulating factor to try and promote tissue repair and healing. Now, neutrophils are a little different. Neutrophils are the very first phagocytic cells to respond. They are not antigen presenting cells, so they just phagocytize like crazy. They just gobble it all up. They are considered the cleaning crew, and they also do not differentiate between a pathogen and cell debris. They are just there. They're just kind of coming in as big garbage men, and they're grabbing everything and cleaning it all. They do have much greater phagocytic ability than macrophages, but their lifespan is very short. And this is because they can actually release strands of DNA to create a net. And they use that net to catch a whole bunch of bacteria at once so they can phagocytize them. So I found this really interesting video that is a time lapse of a macrophage carrying an activated macrophage carrying out phagocytosis, and then a neutrophil carrying out phagocytosis. And in the neutrophil, you might be able to see the actual little kind of net, like the actual trying to release of that net to trap the microorganisms. So here we have, right here, down here at the bottom, we have this phagocytic cell, and it's an activated macrophage. And look at how it's just binding all of these bacteria, and these guys are all coming in through here. I don't think I can write on this, but they're all coming in, and it's just destroying them very quickly. It's consuming tons and tons of these bacteria. You see them forming in the phagosome, and they're just destroyed very rapidly. Now here we have a neutrophil. This is a little bit slower here. The macrophage, when it's in an activated state, is very quick. But you notice this is much smoother here, so it's the same time lapse, just the neutrophil is a little faster in the time lapse. You can see other parts of the cell in here. These are all granules, and you can see that it's kind of moving that net around out here, and it's beginning to take in and create, there we go, and it's starting to take in these cells and break them down. So phagocytosis is a really fascinating process. This is the neutrophil after phagocytosis of it. Here we have another neutrophil phagocytizing. This is fluorescently labeled so that when certain cytokines are produced, they're activated. So there we have phagocytosis. So just to review real quick, make sure that you know the function and all of the steps in phagocytosis, which cells respond, which ones are more phagocytic than others, how when macrophages get differentiated, what do they get differentiated into, and the functions of those. Make sure that you go through a lot of these questions because it will help with, of course, with the exam. And then what is the neutrophil extracellular trap, the nets? Briefly discuss that in that last slide and what are they made of. So I hope you enjoyed this lecture. I will see you guys in part 8. I think we're talking about fever next.